busy week with lots of visitors. We appreciate you taking out the time. We're interviewing a candidate for the Center for Glial Biology at the VTCRI, as many of you know. And I'm really excited to introduce Thomas Papouin, who is visiting us from Tufts in Boston. Thomas is a native of France. He did his undergraduate and graduate work in France. He got his Ph.D. in Bordeaux in 2011 with Stéphane Ollier, probably for us glial biologists, the most recognized name in glial biology, and not just France, but Europe. He went to join Phil Hayden, another very recognized name in glial biology, at Tufts, where he was a postdoc for about 5 years, and the last year he's been a research assistant professor. Now, we all like to make great discoveries as students, and most of us, look back thinking, well, I probably contributed something to that. But very few people actually can really say that they've made seminal discoveries as a graduate student, and Thomas is one of those. He had for his thesis work a Nature paper followed by a Cell paper, and what he was studying was the role of D-Serine as a co-agonist of the NMDA receptor. This was a concept that was discovered a few years earlier, that the NMDA receptor has these modulatory sites, but what were the endogenous modulators and where they were coming from was really not known at the time. So he showed that D-Serine was released by astrocytes, is essential to drive LTP in vivo, and that was the substrate of his Nature paper, really suggesting that while the NMDA receptor can be regulated in multiple ways, astrocytes play a major role. They have the enzyme that converts, it's a racemase that converts the L form to the D form of serine that actually acts at that site. Now, there is the other modulator, which is the glycine site, and he went on to show in the Cell paper that extrasynaptic versus synaptic sites, NMDA receptors, were actually differentially modulated by D-Serine, the synaptic sites, versus the glycine modulating the extrasynaptic NMDA receptors. So very elegant, very comprehensive Cell paper. So this has been really propelling the field of glial neuronal interaction, showing how astrocytic release modulators can profoundly influence the function of an NMDA receptor in a way that makes a lot of sense. And then he joined Phil Hayden's group really to take this into very complex behavioral models. And Phil Hayden is studying sleep cycle, awakeness, if you will, and the role of adenosine in the process. And over the last 4 or 5 years, Thomas has really put together a tour de force study that we just published in Neuron a couple of months ago that I'm sure he's going to talk about today, where he shows that it's alpha-7 nicotinic receptors on astrocytes that via the release of, via the modulation of the modulatory sites on NMDA receptors are really instrumental in driving wakefulness. So over his short academic career, he's published 12 papers, 8 as first author, and they're in Nature, Neuron, Nature Neuroscience, PNAS, the list goes, Cell. I mean, really, really high impact work. So we're very fortunate to have him here. He's been supported by a Human Frontier Science Program grant. These are very competitive grants to get. He has an NIH grant that he's written, and that is undergoing review. So he's formulated some interesting hypotheses for his future career that he's going to share with us tomorrow at the Chalk Talk. So for the moment, without further ado, on the control of NMDA receptors by Astrocyte-Derived D-Serine.
Thank you. Oh, that's loud. Thank you for the introduction. That was definitely worth coming just to hear that. So my introduction is probably going to seem long, but I want to talk you through what I did a little bit doing my PhD and my early postdoc. It's going to have 3 main parts. I'm going to first talk to you about the coagonist binding site of NMDA receptors and why it is so important for the regulation of the activity of the receptor. Then I will talk to you about D-Serine, which is the compound that the ligand that binds this site. And then I will go into the concept that I don't like so much of tripartite synapse that explains or tries to explain how the availability of D-Serine is regulated. If you have any issue with my accent anytime during the talk, please just stop me. So NMDA receptors, you're all familiar with NMDA receptors for the very simple fact that it is the most investigated receptor in neuroscience. There's about 1,600 publications a year. That means about 5 to 6 a day. In other words, you leave the lab on Friday night, you come back on Mondays, there's been like 11 publications. It's just impossible to keep up. One of the reasons it is so heavily investigated is probably because of the fact that it is permeable to calcium. And that makes it involved not only in synaptic transmission, but also in a lot of processes that involve intracellular signaling, such as synaptic maturation, synaptic plasticity, and also excitotoxicity when you have an overload of calcium in the postsynaptic element. Besides the fact that it is so heavily investigated, there is a major aspect of its regulation that has been completely undermined, and it is the fact that in addition to glutamate, it requires the binding of a so-called coagonist on a distinct and dedicated coagonist binding site. And this is absolutely important for the activation of the receptor, to the point that if you do not provide agonist to the coagonist binding site, it doesn't matter how much glutamate you give to the receptor, it will never open. So this is not a modulatory site, this is just another agonist site. And it was very well illustrated by the seminal study from Johnson and Asher in '86, where they find that if they record NMDA responses while applying just NMDA or just glutamate, they basically have nothing at all. This is not sufficient to activate the receptor on its own, which is already kind of a little bugging for an NMDA receptor. And they found that they need to provide both the agonist of what's usually called the main agonist binding site and the agonist of the other agonist binding site for the receptor to open. Okay? Now, from this seminal study, we have doomed this site, the coagonist binding site or the glycine binding site, which is very unfortunate for two reasons. First, because we found out that in most cases, glycine doesn't actually bind this site, at least in the tissue. And also because the term coagonist binding site makes you feel like this is something secondary to the activation of the receptor, something completely auxiliary, like a form of modulation. And it really isn't. In fact, if you had to decide which of these two sites is the most important, it would definitely be the coagonist binding site for two reasons. First, because it is on the gluN1 subunit of the receptor, and that is the obligatory subunit of the receptor. In other words, you will never find an NMDA receptor that doesn't have two gluN1 subunits, and therefore that doesn't have two coagonist binding sites. The glutamate binding site, on the other hand, is on the gluN2 subunit, which is very optional. So optional, in fact, that we do have NMDA receptors that are composed of gluN1 and gluN3 subunits. Therefore, they do not have a gluN2 subunit. Therefore, they do not have a glutamate binding site. Therefore, they are glutamate receptors that do not bind glutamate. Okay? They do bind glycine, though, and D-Serine, with outstanding affinities, by the way. I don't have time to talk about this. The other reason why this site is extremely important, and more important than the glutamate binding site, is because from the point of view of glutamate binding, any synaptic transmission event is fully saturating for this site. And we've known that since 1998. There's about 3.2, 3.6 millimolars of glutamate released in the synaptic cleft during the few hundreds of microseconds after the fusion of a single vesicle, which is about 10,000 times above the EC50 of these sites. Okay? So in other words, if you're looking at a population of NMDA receptors, the only way you can decide how many of these receptors are going to open, truly be activated during synaptic transmission, it is not through glutamate, it is through the modulation of how much coagonist you provide for these sites. Right? So of course the question becomes, first of all, who is the compound binding this site? And contrary to what we thought for a long time, in most cases, and at least for the purpose of this talk, it is not glycine, it is D-Serine. And we showed that, we and others, showed that in multiple ways, and the way we showed it was by using enzymatic scavengers that would degrade either endogenous D-Serine or endogenous glycine. And we found that when we get rid, the best we can, of glycine in the tissue, NMDA function is completely unaltered. But if we deplete the best we can D-Serine with this enzyme, then we lose a large portion of NMDA receptor activation. Right? The reason why people like me got extremely excited about this is because we think that D-Serine comes from astrocytes. So this is the very first immunohistochemistry ever performed against D-Serine, and you can find, like authors concluded, that it is mostly found, if not exclusively found, in glia. And this is a more recent EM picture from the group of Andrea Volterra in Switzerland, where they found that indeed D-Serine, you're looking at the small immunogold particles here. D-Serine is present in the astrocyte process that contacts the synapse, and it's mostly present in what we call synaptic-like microvesicles, which are basically astrocytes vesicles. And a year after that, people from the lab where I did my Ph.D. were actually able to pull down those vesicles with a technique that actually Michel Olson is probably very familiar with, magnetic microbeads. And what they found is that these vesicles indeed contain D-Serine, but not only that, they also contain a transporter that seems selectively involved in the uptake and upload of D-Serine into those vesicles. And cherry on top, these vesicles also contain serine racemase, which we believe is the enzyme that makes D-Serine from L-serine. In other words, you have an entire system here in astrocytes to provide the coagonist of NMD receptors, to synthesize it and provide it to NMD receptors, right? In other words, you have a system in which the regulation of the most important, the most investigated receptor in neuroscience is actually in the hands of astrocytes. And that's indeed what we showed back in 2010 with the group of Dmitry Ryzakov at UCL. What we did is we patched an astrocyte in the hippocampus, and in this patch pipette, and most of you here doing EFIs probably know that, in this patch pipette, you can put whatever you want. You can put an antagonist of serine racemase, you can put tetanus toxin to prevent the fusion of these vesicles, or you can put what we used here, a calcium clamp solution that is going to maintain calcium in astrocytes at a resting level. And we know this is going to prevent the calcium-dependent release of vesicles. And in the meantime, we also record NMD responses with another pipette in the territory, or more or less, in the territory of the given astrocytes. And we found that as soon as we start dialyzing the solution into the astrocytes, we initiate a rundown of NMD responses, which can be completely reversed by providing exogenous D-Serine, or can be completely blocked by having D-Serine in the bath to begin with. In other words, as soon as you start preventing the release of D-Serine-containing vesicles from astrocytes, then you start losing the activation of NMD receptors on neurons in the territory of the astrocytes. Since most people who work on NMD receptors only care about synaptic plasticity, we also showed that obviously the exact same technique, the exact same approach enabled us to completely abolish the expression of LTP in the territory of this given astrocyte, but not the neighboring astrocyte. So this was a quick cruise through 30 years of research, but basically the take-home message at this point is that the control of NMD receptors that you're all very familiar with is actually not at all in the hands of neurons through the release of glutamate. It is actually entirely in the hands of a glial cell, astrocytes, through the release of D-Serine, and this is probably why we were able to publish that in Nature back then. However, the question I started with, which is how do you regulate how much D-Serine is available, completely remains, because we still don't know how exactly and when and why astrocytes release D-Serine. So the question I started with remains. And to try and tackle this, you have to look into a concept that I somewhat dislike, called the concept of tripartite synapse, which tells you how those three elements, the astrocyte, the presynaptic neuron, and the postsynaptic neuron, interact together at synapses. So this concept is primarily an anatomical concept in that it tells you how this very complex, convoluted, and intricated astrocyte process, which is not really a process, it's actually more like a sponge-like thing, is interacting with neuronal elements, and especially synapses. Okay? You probably know very well this picture from, it was from Mark Witcher, who's working here. It is also fundamentally a functional concept that is trying to tell us that we can no longer see synapses as only made of a presynaptic and a postsynaptic element, but also comprising of the astrocyte process that unwraps those two and can listen to what's happening at the synapse and also release transmitters called gliotransmitters to impact the function of synapses, right? And we have accumulated a number of examples of gliotransmitters that astrocyte can release. And of course, D-Serine is one of them. But this is not the only one. We've accumulated many, many examples to the fact that it simply does not make sense anymore. And I do not mean it in a judgmental way. I mean it in the most primordial way, as in, after 20 years of working with this concept, we have not yet been able to make sense of what it is exactly that astrocytes do at synapses through the release of any of these transmitters. We have not been able to show that the release of adenosine, for example, is absolutely primordial for synaptic computation and or behavior. And you will not, in fact, find a single paper telling you that through any of these particular pathways, astrocytes are absolutely necessary in a form of behavior, right? At least that's my take on it. And the reason is because of the way we think of this entire concept. We think of it as, and those are two words you would see a lot in reviews about a tripartite synapse, we think of it as a regulation or a control of synaptic transmission by astrocytes. But neither of these work. The concept of regulation, at least in French, I'm not sure in English, but at least in French, the concept of regulation entails a very strict and unambiguous relationship between a variable that would go out of range and an output effector system that would bring back the variable within its physiological range. That is the notion of regulation, at least in French. So in this case, you have to assume that the variable is synaptic transmission, so glutamate release. But you see right away that the same glutamate release can trigger different pathways that would positively or negatively impact glutamate release. So this is simply not a regulatory loop. So okay, maybe French people are just a pain, and maybe by regulation we meant really just control. But it doesn't work either, because the notion of control intrinsically comes with the notion of timing. You want to be quick at controlling what you're supposed to control. But in this case, what we quantitatively know from the signaling machinery of astrocytes is that they're slow. They are very slow cells. They're in fact the slowest secretory cell of the nervous system. And this is an example, quite a dramatic example, but an example from the literature, where you're looking at the calcium activity in the postsynaptic neuron and the neighboring astrocytes in response to a train of presynaptic activity. And if you look at the time scale, the astrocyte takes something like 10 seconds to react. So whatever it is in this case that astrocyte released, the signal that it was meant to control is long gone, all right? So I don't think any of these two concepts really work. However, they tell us very interesting things about what exactly it is that's going wrong. And I think what's going wrong is that we see this trio as some kind of a closed loop, completely blind to whatever is outside. It's this kind of an unhealthy ménage à trois, if you will, where we know at least we want to think that astrocytes are important in this concept, otherwise I wouldn't even be here talking about it. We have many proofs that astrocytes can impact synapses through the release of transmitters. So we have to assume that what's wrong is the first step here, thinking that they take instructions from synapses themselves. And in fact, this is exactly what working on D-Serine for 10 years has taught me. Evolutionary speaking, from an evolutionary point of view, there is absolutely no reason why you would have an entirely distinct cell type with its own physiology, its own anatomy, in charge of synthesizing and releasing one of the two agonists of NMDA receptors, unless you can do that completely independently than what's happening at the synapse itself. All right? So my idea when I joined the Hayden lab was to try and open up this concept into something more linear, where astrocytes would get their information from external signals completely independently than what's happening at synapses, then crunch this information, integrate it, and feed it to synapses in a way they understand, which means through gliotransmission. All right? So the question that's been driving my research over the past few years, and will continue to drive it for the next probably 10 years at the very least, is, I'm going to come back to that just later, is whether astrocytes are following brain states rather than synapses. And the reason is because among the many things that astrocyte can listen to, it's become very clear that they're really good at listening to neuromodulators, such as norepinephrine and acetylcholine. And this makes this concept even more interesting and exciting because neuromodulators are typically released during given brain states, which would make astrocytes able to listen to brain states or brain context, if you will, and use that to tune synapses, which I think is a more interesting concept than this very closed loop. I was going to say ménage à trois, but so this is the question that's been driving my research lately, and I went to the Hayden lab to try and address this question, especially because the Hayden lab came up with the concept of tripartite synapse to begin with. Of course, this is way too much work for a single postdoc. I wasn't going to answer that question as a postdoc, so I went back to something I knew better, which is D-Serine, and asked these questions specifically in the field of D-Serine, trying to understand now what is regulating D-Serine availability by making the hypothesis that it would be brain states through neuromodulators. So the question I tried to answer was, is D-Serine availability brain state dependent? I didn't quite know what it really meant when I studied my postdoc. I had a very vague idea of what it meant, but I assumed that if something varies across brain states, then it definitely varies across the sleep-wake cycle. And since the lab had a history of working on the sleep-wake cycle, I figured I'm going to probably try looking throughout the sleep-wake cycle of mice. Yes?
A quick question on your previous slide. I'm sure you'll get into the details, but when you state the question, is D-Serine availability for the state? You didn't say D-Serine release, but availability. I'm sure
Yes.
you mean that. So right off the bat, when you started with the notion from a cell biological, biochemical point of view, that it would be regulation of availability, is that literally?
Yeah, that's literally what I meant, because you could be looking at the release, the synthesis, or the uptake, and there's many aspects of these, many of these aspects in D-Serine physiology that we don't know about. The uptake of D-Serine by some supposedly transporters is really obscure so far. Even the synthesis of D-Serine is not as straightforward as some people would like it to be. So I meant availability in the widest sense, really in terms of how much is available to NMD receptors. So the first thing I did, since this was a very challenging question, I decided to go use the simplest approach that's possible, which I think is, by the way, a good advice for the students here. When you have a question that seems a little too challenging, take the easiest approach you can. That's going to eliminate a lot of complications. So we obtained hippocampal slices from mice at different times of the day, and we simply recorded NMD responses in these slices for the simple reason that NMD receptors are the best D-Serine sensor you could imagine. They're at synapses. They have a great affinity for D-Serine that's just perfect. And then we applied, to understand how much D-Serine they already have available, we applied extra exogenous D-Serine to try and saturate the receptor. This would give you this increase in the response. From that, you can calculate what we call the saturation index, which is really just how much occupied were the receptors originally, right? So I'm going to show you two things throughout my presentations, either the effect of D-Serine itself, how much it potentiated the receptor, or what we call the saturation index that we calculated from that increase, right? And it's a very simple equation. And before I forget, for those of you who are not familiar with the ZT time scale, the Zeitgeber time scale, it really is just a 24 hours period with the origin at the onset of the light phase. So basically ZT0 is when the light turns on, and it's usually 7 a.m. In the animal facility. And ZT1 is 1 hour later, etc. So doing that, what we found was that the effect of D-Serine application on NMD responses, in other words, this little increase here, was extremely dependent on the time at which slices were obtained. So when we take a slice at ZT0, which means after the mice have been awake during the dark phase, just before they go to sleep, in other words, we had absolutely no effect of D-Serine application, which told us, those are the raw data, which told us right away that at this time of the day, NMD receptors were fully saturated with D-Serine in the slice. Now, if we do the exact same thing in slices that are obtained in the light phase, when mice are mostly sleeping, or even at the very onset of the dark phase when they just woke up, we had a 25 to 30% increase induced by D-Serine on NMD responses, which told us that the receptors were not saturated. There was not enough coagonist in the slice to fully saturate the coagonist binding site of the receptor. So to try and understand what mechanism we were dealing with, we wanted to know if these oscillations were circadian, as in really dictated by the time of day at which the slices were obtained, or wakefulness dependent, as in due to the fact that mice are mostly active during the dark phase and mostly sleeping during the light phase. And there is a very easy way to distinguish between those two possible mechanisms. It's to simply do what we call a sleep deprivation. When you take the mouse at ZT0, when it would like to go to sleep, and you maintain it, you keep it awake for a few hours. Now, if the oscillations we saw are time of day dependent, then this is not going to change at all, because the time of day per se didn't change. But if the oscillations are driven by wakefulness, then we should be able to shift them to the right just because we're keeping the mouse awake. So that's what we did, and that's exactly what we found, actually, that doing 2 hours sleep deprivation was completely enough to maintain a full saturation of NMD receptors compared to mice that were able to sleep during those 2 hours. And then we thought to do the exact same thing during the dark phase, especially because we were kind of puzzled why it would take so long for D-Serine to accumulate, for NMD receptors to reach full saturation during the dark phase, until we realized that, well, in labs, mice don't have much to do in their little cage. Okay? So they don't actually spend so much time awake during the dark phase. They are very active at the very beginning, but then they take a lot of naps, basically, because they don't have much to do. So we wondered, can we boost their activity by providing some kind of enriched environment? And we found that we can. So these are actually two different experiments, but if you just give a wheel to a mouse in its cage, it will spend the first 6 hours straight in the dark phase just being active and not going to sleep at all. And we found that this was indeed sufficient to increase, to accelerate the buildup of an NMD receptor saturation, especially if you look at 6 hours within the dark phase. So after those 6 hours of playing with the wheel, we now found that NMD receptors were fully saturated as opposed to when mice are kept in their home cage without a wheel. So I tried to be very careful here talking about the coagonist binding site of NMD receptors and its occupancy and blah, blah, blah. Because at this point, we didn't actually have any proof that this was just fluctuations in D-Serine levels. All right. So we did find there was fluctuations in the occupancy of the site of NMD receptors, but then we wanted to check whether this was indeed due to D-Serine oscillations. We used to do that the very similar approach. We obtained slices at different times of the day, but then we used basically amperometric biosensors, which are simply a platinum wire with a D-amino acid oxidase coating, which is the enzyme that degrades D-Serine. And in other words, for each molecule of D-Serine degraded, you will get 2 electrons. This is what the sensors look like. They're basically just a pipette with the coating of DAO that you can see here. And they give you a very nice linear readout, electronic readout for increasing concentrations of D-Serine. So again, what we did was obtain slices, hippocampal slices from mice at different times of the day. We incubated these slices in tiny amounts of ACSF. And then we ran this, what we call conditioned medium, on the sensors. This is the type of signal that we get. And we found that indeed, in the conditioned medium obtained from ZT0 slices, so slices obtained at the end of the dark phase when mice are active, we found more D-Serine than in conditioned medium obtained from ZT6 slices when mice were sleeping. And this is normalized to the amount of tissue incubated, and you can see that the amounts of D-Serine, again, match really well the saturation index of NMD receptors. Yes? Yeah, that's a very good question. We tried and we found that there was several issues. First of all is this coating here is extremely fragile. So many times when we used it, like we poked the slice, the coating would come off, which was a bummer because these cost $1,000 each. And also we found that if we introduce the sensor in the slice, we're just going to crush the tissue. And it just happens that 99.6% of D-Serine is intracellular. So we would have a huge spike of D-Serine, and then we wouldn't know which of that is really just extracellular versus from the cells that just exploded. This is about 50 microns. 50 microns? So from there, we concluded that there were indeed daily fluctuations in how much D-Serine is available to NMD receptors in slices. The next question was, of course, is it an artifact from slices or is it something that really happens in vivo? So we moved to doing in vivo microdialysis where you can sample the extracellular fluid basically from the hippocampus, coupled to HPLC to detect D-Serine and L-serine. But on top of doing that, we also monitored mice activity either through EEG and EMG recording or just video monitoring. And you can see here that both give you really the same readout of mice activity. And we found, well, first we found that the amounts of D and L serine we detected in the brain was very, very similar to what most people find in the literature. But then when we started plotting for each mouse how much D-serine we found versus how active the mouse was during dialysis, we found that there was a very neat regression telling us that the more the mouse was awake during microdialysis, the more D-serine we collected. And for those of you who are familiar with NMD receptors, these concentrations span the entire range of NMD receptor sensitivity to D-serine, which was pretty cool too. Interestingly, this regression only existed for D-serine. We didn't look at every single spike we get, but at least it didn't happen for L-serine, which is an important control because it tells you that this is not just the access to the extracellular fluid that changes throughout wakefulness, right? So we concluded at this point that D-serine availability is indeed dependent on wakefulness in vitro and in vivo. We then wanted to know how significant is that? Can that impact behavior? So we're looking at something that impacts NMD receptor activity in the hippocampus. So obviously we thought of doing a task, a behavioral task, that is NMDA dependent and hippocampus dependent. So we opted for contextual fear memory. For those of you who are not familiar with the test, the mice spend 3 minutes in this little cage and then they get shocked. And they're very quickly going to learn that the environment is aversive. And you can measure that by measuring how much they freeze. We found that regardless of the time of day at which we train the mice, they all learn pretty much the same way. You see they start freezing after the shocks. But then when we put them back in the environment 24 hours later to assess their memory, we found that the mice that were trained and tested as T0, when we know D-serine is the highest, had a much better performance than mice that were trained and tested as T6 in the light phase when we know D-serine is low. Okay. There was, of course, a lot of control experiments built in this one to check, first of all, that these effects were not due to the fact that mice have more or less sleep time just after training. We also checked that this was context dependent, that there was no impairment in the acquisition of the task per se. But most importantly, when we went back to doing these simple experiments with injections of D-serine, we found that injecting D-serine in mice prior to training at T0, when we think D-serine is already high, didn't improve their memory, their performance, in line with the fact that D-serine is already saturating at this time in the hippocampus. But we found that injecting D-serine in mice at T6 during the light phase prior to training could improve their performance, again in line with the idea that D-serine is not fully saturating at this time to NMD receptors. So conclusion of this first part are that we found oscillations in NMD receptor activation throughout the sleep-wake cycle. They correspond to oscillations in D-serine levels. Those are driven by wakefulness. They also happen in vivo, and they are significant enough that they impact behavior. So we were getting slightly closer to understanding what drives D-serine oscillations, because now we know that we're looking for wakefulness-dependent clues, a wakefulness-dependent signal. Yep. No, there were IP injections. Yeah, we could have done local injections, which I would have expected to do the exact same, lead to the exact same effect. We just did IP injections. Yep.
So you showed that just activity increases in D-serine. But you don't know if it's binary. Either the mouse is active or not active. So does the amount of activity
Oh, that's a great question. We haven't answered that in that study paper. That's something I'm interested in looking at later. We don't have the time resolution with microdialysis to say exactly what part of activity is really driving D-serine release, if it's a particular brain state, or if it's really the locomotion activity, or the fact that they're awake. We haven't really tested for that. So that's a great question. I don't really have an answer yet. It's just the more the mouse has been awake at the time you collect D-serine, the more you're going to find D-serine. And if the mouse has been mostly sleeping or being inactive, then there is less D-serine. So we thought we had narrowed down quite a bit what were the clues that we were looking for that drive D-serine release or availability. The fact that is actually that we didn't at all, because there is a lot of things that vary with wakefulness. You can think of serotonin, norepinephrine, orexin, acetylcholine, and that's just naming a few neuromodulators. We did, however, get a break from looking at schizophrenia. And I know it seems like far-fetched, but it's really not. There are two main hypotheses in the etiology and the treatment of schizophrenia. The first one is, like it is stated here, the glutamate hypothesis. And this is mostly based on the idea that there is a hyperfunction of NMDA receptors. Now, like I just told you at the beginning of the talk, hyperfunction of NMDA receptors probably means a hyperfunction of the coagonist binding site. And this is indeed the case, either because there is a mutation in the coagonist binding site, or because, and this is now very notorious, schizophrenic patients have low D-serine levels in their brain, in their plasma, in their CSF. The other major hypothesis of schizophrenia, and you probably know that, is what's called the cholinergic hypothesis. And it is very well illustrated by just looking at the prevalence of smokers within the schizophrenic populations. It's about 75%, which is 3 to 4 times higher than the average population in the US. That wouldn't be true in France, but in the US, that's about 4 times difference. And so we think this is an indication that smoking is some kind of a self-medication where patients unconsciously try to boost their nicotinic activity. And to make a very long story quite short, among all of the nicotinic receptors, the ones that have been especially hot lately, especially in clinical trials, are the alpha-7 nicotinic receptors. Not necessarily because they are directly linked to schizophrenia, but because there's been a lot of trials that showed that targeting these receptors restore completely or partially the cognitive aspects of schizophrenia. So we were simply wondering if maybe these two major and seemingly distinct hypotheses of schizophrenia could be somehow linked, in particular via D-serine. And it seemed like indeed acetylcholine and alpha-7 receptors in particular were very good candidates because acetylcholine varies throughout the sleep-wake cycle. It is high during quiet wakefulness and even higher during active wakefulness compared to sleep. We know as well that astrocytes are able to listen to acetylcholine and respond to acetylcholine, especially through alpha-7 nicotinic receptors. We even have evidence from cells derived from astrocytomas, actually, that challenging them with acetylcholine can boost the synthesis and the release of D-serine. And we also have evidence that mice that are knocked out for alpha-7 nicotinic receptors have deficits in NMDA receptor function. So all of this together really made us believe that it was very likely that acetylcholine through alpha-7 nicotinic receptors was what drives D-serine oscillations throughout wakefulness. To test this, it looks brutal, but it's actually very simple. To test this, we went back to take slices from the hippocampus during the light phase, when we know that D-serine is low and low enough that the receptors are not saturated. And then the game was simply apply anything we found in the lab to try and improve this saturation level. Yeah, because things didn't happen the way I'm telling you. The first thing we tried was actually applying norepinephrine because it's a very sexy target these days in the field of glial cells. And it just happened that norepinephrine had no effect whatsoever on the saturation index. Then we tried carbachol, which is an agonist of all cholinergic receptors, and found that just within 20 or 30 minutes, that was completely enough to fully saturate the coagonist binding site of NMDA receptors. So that was exciting, and from there the game became let's try and block the effect of carbachol with various antagonists, right? So the first thing we tried was obviously atropine, which is an antagonist of muscarinic receptors. And surprisingly, it did not block the effect of carbachol. And that was actually very surprising because for those of you who are familiar with astrocytes, we think that astrocytes activity is mainly modulated by metabotropic receptors. So we thought this was going to be a very good candidate, but it turned out not to be. So then we tried to block the effect of carbachol with this antagonist, which is an antagonist of alpha-4 beta-2 nicotinic receptors, which are the most expressed nicotinic receptors in C57 black cyst mice. And again, it didn't do anything at all. And then we tried MLA, which is the antagonist of alpha-7 nicotinic receptors, and found that this antagonist was completely able to prevent the effect of carbachol itself. And then if we applied a selective agonist of alpha-7 receptors, we were able to totally replicate the effect of carbachol. Right? So in other words, it looked as if stimulating selectively alpha-7 nicotinic receptors was sufficient to elevate D-Serine levels in the slices. So the next question was obviously, is it also necessary to have high levels of D-Serine? And we had a very simple way of testing that by taking slices at the T0, so at the very end of the dark phase, after mice have been active. And when we know that D-Serine is naturally high in slices, and we incubated these slices with MLA, the antagonist of alpha-7 receptors, and found that incubating these slices with this antagonist for 45 to 60 minutes was sufficient to completely desaturate NMD receptors coagonist sites. All right? So in other words, stimulating alpha-7 receptors can improve D-Serine levels in the slice, and blocking alpha-7 receptors can decrease D-Serine in the slice. Of course, the next question was, where exactly does acetylcholine come from in the hippocampus? And this wasn't actually a trivial question. The vast majority of cholinergic innervation in the hippocampus comes from a discrete brain region called the medial septum that bursts with trains of 5 hertz during wakefulness. And what's very interesting here, I'm not going to read all of this, is that the cholinergic inputs from this region into the hippocampus do not synapse on neurons or astrocytes. 80 to 90% of them make presynaptic boutons with no postsynaptic entity facing them. In other words, we think that the cholinergic projections in the hippocampus are just meant to set what's called a cholinergic tone. In other words, just increase extracellular levels of acetylcholine in the tissue, in the extracellular space, which is great if you want astrocytes to be able to sense acetylcholine levels. So what we did is we took mice that express, well, you can't see really well, but mice that express channel rhodopsin in these particular neurons. And you can see here those beautiful cholinergic neurons in the medial septum and the diagonal vena broca that innervates the hippocampus. And if you look at the hippocampus here, you don't really see neurons. All you see are fibers and in some instances varicosities, so presynaptic cholinergic boutons. And what we did was we recorded NMD responses in these slices for about 30 minutes. And then we stimulated with blue light those neurons. And we found, this is one example, and this is 10 experiments. We found that this was sufficient to cause a long-term enhancement of NMD responses. But the most exciting part in this experiment was that if we then applied D-Serine to try and question how saturated were the receptors at this point, there was no effect at all of providing exogenous D-Serine, indicating that the receptors at this point, after we had stimulated the cholinergic fibers, were completely saturated with D-Serine, even though at this time of day, they were not supposed to be. Right? So we think what's happening here is that simply stimulating with blue light the cholinergic fibers that run through the hippocampus was completely sufficient to provide additional D-Serine to the receptors and therefore boost NMD receptor function. Since we think that this is mediated by alpha-7 nicotinic receptors, the next experiment was obviously to repeat that with MLA in the bat, which is the alpha-7 nicotinic receptor antagonist. And as you can see, this was sufficient to completely block the effect of the light, per se. But at the end of the experiment now, if you try and apply D-Serine you have this nice potentiation that tells you, okay, NMDA receptors were not saturated. There is still room for improvement. They're not maxed out with D-Serine. In other words, when you stimulate the cholinergic fibers but block alpha-7 nicotinic receptors at the same time, you cannot anymore improve D-Serine levels. And then we did a lot of experiments with D-Serine in the bath. I'm just going to skip that. But basically, the take-home message of this part is that by simply playing with alpha-7 nicotinic receptors, we can improve or diminish D-Serine levels in slices, including when we stimulate acetylcholine levels released from the fibers that are in the hippocampus. So at this point, of course, it's about time that I start talking about astrocytes and what exactly is the role of astrocytes in this overall picture. To tackle that question, we used D-Serine mice that we had in the, in the Hayden lab. In these mice, astrocytes express a dominant negative form of VAMP2, which is a snare protein. So in astrocytes in these mice, the vesicles in astrocytes have to compete so much for fusion and for binding and fusion that actually their release is prevented in about 90% of the case. So as soon as we take mice off doxycycline, astrocytes no longer release vesicles, in particular the vesicles that contain D-Serine. And you probably heard a little bit about these mice because they supposedly express the transgene a lot in neurons, and I'm going to let you judge of that by yourself. This is not true at all. You have all of the astrocytes here. Neurons are labeled here with for nu N, and you can see that the EGFP transgene doesn't colocalize with neurons whatsoever. What we did find, though, is that in these mice, the oscillations that we had found initially, oscillations in the effect of D-Serine applications, are completely gone as soon as you put the mice off dox and glial transmission starts to be inhibited. It is not to say that NMDA receptors are completely unsaturated. It just means that the wakefulness-dependent increase in D-Serine availability is inhibited, right? So NMDA receptors are always in presence of the same amount of D-Serine, and you never max them up by having additional D-Serine released during the dark active phase, right? So then we started to wonder, okay, how does that fit in the overall schematic we have in mind? So we went back to these pharmacological experiments where we found that stimulating all cholinergic receptors or stimulating just alpha-7 receptors was sufficient to boost D-Serine levels, and found that this was completely inefficient in the D-Serine mice in which the release of vesicles is impaired to begin with. And it doesn't seem like much, but what that tells you is somehow astrocytes are in between acetylcholine activity and NMDA receptors. Especially, they're in between the stimulation of alpha-7 receptors and D-Serine release. So the most parsimonious hypothesis was obviously that alpha-7 receptors were actually located on astrocytes. Okay? And to test that, we obtained mice that are fluxed for the exon 4 of alpha-7 nicotinic receptors from Kelly Dinelli in Texas. And we injected these mice with an AAV5 GFAP Cre virus, which would lead to the deletion of alpha-7 nicotinic receptors exclusively in astrocytes. And here you have an immuno of the expression of the GFP tag of the virus. We found that about 80% of all astrocytes in the CA1 express the virus, so or recombined for alpha-7. And we found that this virus was really only targeting astrocytes and no neurons. We had only 2 cells that we were not sure about. One of them was microglia, and with the other one we never found out. In these mice injected with the Cre virus, we found that the oscillations, pretty much like in the D-Serine mice, were mostly gone. Quantitatively, it's not stunning, but qualitatively, this is really what we were expecting, as in you no longer have the wakefulness-dependent accumulation of D-Serine, which is retained, however, in the control ligaments injected with the virus. Okay, so in these mice, in knockout mice, NMDA receptor saturation remains fairly low and constant throughout the day. So if you take all of this together, obviously, sorry, if we do the exact same thing in neurons where we inject an AAV9 synapsin Cre virus pre and post synaptically, the oscillations are completely intact. Obviously, both in the knockout animals and in the control lineamates, telling us that knocking out alpha-7 receptors in neurons doesn't impair D-Serine oscillations throughout the day. So the take-home message was this. What we think is happening is that astrocytes are detecting and listening cholinergic levels, acetylcholine levels, that are set by the activity of this remote brain region in a wakefulness-dependent manner, and use this information through alpha-7 nicotinic receptors to determine how much D-Serine should be released to NMD receptors at synapses. And by doing so, they're enabled to match the capability of synapses to basically make synaptic memory to the ongoing brain state, which in this case is wakefulness. But besides D-Serine, because I know most of you probably couldn't care less about D-Serine, besides D-Serine, what we think this shows is that this might be the first actual demonstration that what astrocytes do is not listen to what's happening at synapses, but rather listen to the brain state-dependent activity of a given brain region, a remote brain region, through the release of neuromodulators, and use that information to properly shape the properties of synapses within the territory, and in this way, match the properties of synapses to the ongoing brain context. And this is a concept we decided to call contextual guidance, in which astrocytes listen to brain states to give some kind of a guidance to synapses. So how is that a step up from the original concept of the tripartite synapse? Well, it is in many ways. One of them is because now instead of going fishing in trying to find out what is the role of astrocytes in behavior, for example, you can actually completely predict what is going to be their role and when. So, for example, we know from schizophrenia that impairments in cholinergic signaling or NMDA receptor signaling leads to a set of behaviors such as impaired working memory, impaired spatial memory, impaired preference for social novelty, and so on. We just showed that basically astrocytes merge cholinergic signaling and NMDA signaling. So you would assume that astrocytes are precisely involved in supporting these particular cognitive functions, and therefore you would assume that if we disrupt this link, for example, by knocking out alpha-7 receptors in astrocytes, this would lead to a very similar set of behavioral deficits. So to test for that, what we've done so far is we generated cell-specific knockouts to increase our throughput. We generated mice that are knocked out for alpha-7 receptors in neurons, or mice that are knocked out for alpha-7 receptors in astrocytes, in blue or in red. We found, luckily, that the neuronal knockouts have no impairments in D-Serine levels, but the astrocyte knockouts have quite a bit of D-Serine depletion. And by the way, this is the strongest effect I've ever seen in D-Serine since the generation of the serine racemase knockout mice that lack D-Serine altogether. And then we started using these mice for behavior. And we started with spatial memory test, a fear conditioning test. We found that the astrocytic knockout have no real impairment for learning, though there's probably something here, but it didn't turn out to be significant. But then when you test their memory 24 hours later, they have a very strong impairment. It's about 45% decrease, which is exactly what we expected from these predictions. What was interesting, too, is that if we if we put mice in a neutral context the next day, we did find that they freeze a little bit, they freeze quite a bit, and actually they freeze, in fact, quite a lot compared to what they were freezing in the fearful environment. So then when we did a ratio between those two, what we realized is that the astrocyte knockout mice have impairment in identifying the safety versus fear cues, which is also something we had predicted from just looking at schizophrenia. Very satisfyingly, what we have so far is that the mice that are knocked out for alpha-7 neurons have no impairment whatsoever. And all of these experiments were done blind, by the way. So they have no impairment at all. Then what we did lately, I'm not going to show you all of this because this is still ongoing, but we looked for possible impairments in the preference for social novelty, which is actually a very simple test. It really just relies on the fact that a mouse will preferentially go interact with a new mouse rather than a mouse that it's already familiar with. And the first step of the test is by simply testing the sociability. So you allow your mouse to either explore a chamber where there is an object or a chamber where there is another mouse. And as you can see, independently of the genotype, all mice spend more time exploring this chamber than that chamber, which makes a lot of sense. And this is also something we had predicted from just looking at schizophrenia. But now, as soon as you introduce another novel mouse in that chamber, the control mice and the neuronal knockout mice tend to explore more this chamber, but not our astrocyte knockout mice, which again is something we had predicted from schizophrenia, and this is called a lack of preference for social novelty. So I could go on like this with a lot of behaviors that we're running right now, but basically by simply looking at this contextual guidance type of concept, you can now completely predict what are the behaviors that what are the behaviors that astrocytes are involved in. And not only that, but you can also predict when are astrocytes involved in these behaviors, because this is obviously going to be context dependent. So in this case, this is going to be wakefulness dependent. So you would expect that all of these impairments are going to be completely gone if you take the mice during the light phase when acetylcholine levels are not so high and this pathway is not really turned on yet. And I'm not going to show it to you because it's preliminary, but so far this is indeed the case. In other words, all of those disruptions in the behavior and cognition are gone if you take mice in a different brain context. Something else I'm obviously very interested in is looking at the effect of alpha-7 nicotinic modulators in the treatment of schizophrenia. Like I said very briefly, those modulators, alpha-7 modulators, make up more than half of the trials currently going on in trying to treat the cholinergic aspect of cognitive deficit in schizophrenic patients. And what I showed you is that basically alpha-7 receptors seem to merge these two aspects of schizophrenia. So the question we want to answer is, do these compounds, such as EVP, which was designed by Fralin Pharmaceutical and is the only compound that made it to phase 3 clinical trial, do they act through astrocytes specifically? And, well, I don't have any data yet to show, but the only thing we found is we are able to increase D-Serine levels in mice by simply injecting one dose of these alpha-7 nicotinic receptors modulators, which indeed suggests to us that they might very well achieve their pro-cognitive effect via astrocytes. All right, so I'm going to end up by thanking, of course, all of the lab. I actually hate this picture because this makes you believe that this is Phil Hayden, but this is, in fact, our lab manager, Bob. This is Phil. This is Jackie, who did a lot of virus injections for the Neuron paper. And this is Michaela, a grad student that I'm working with. She decided to take on all of the microdialysis approaches. Thank you to the institution and fundings. And if you slept during my talk and only have 5 minutes, I invite you to go check our video abstract. And I'll take your questions. So no, we found that if we inject D-Serine during the dark phase, it doesn't improve memory because receptors are already saturated with D-Serine. Yes.
So, of course, acetylcholine is a well-established neuromodulator for plasticity in places like our occipital cortex.
Yes.
Right, in the corticoplasticity. And there it's been demonstrated that presynaptic NMDA receptors release. And I'm kind of wondering how a presynaptic NMDA receptor would fit into the model in this case, if at all.
So in the CA3, CA1 synapses, there is no like convincing proof that there's any NMDA receptors on the presynaptic buton. But there's a lot of evidence that they are in the dentate gyrus, for example. This is notably the group of Andrea Volterra who showed that. You could totally imagine that this would also improve the activity of these receptors and therefore further modulate presynaptic release, especially by astrocytes, because we know astrocytes are involved in this schematic. And that would actually be very interesting because what this group has showed is that presynaptic NMDA receptors in the dentate gyrus are regulated by astrocytes that release glutamate on them. So in this case, they would release glutamate and D-Serine, which will obviously mean that you must have two distinct separate pathways, and it would be a great place to go look to understand how a given neuromodulator couples to a given gliotransmitter pathway. But yeah, you would expect that this also modulates a lot synaptic plasticity, short-term and long-term. Yeah, this is a very good point. Within the hippocampus, it's hard to tell because it's really just setting a tone, a cholinergic tone. But what I'm really interested in is looking at other brain regions where we know that acetylcholine comes from another nucleus. So in the hippocampus, it comes from the medial septum. But if you look at the cortex, for example, it comes from the nucleus basalis of Maynard. So you would expect that the release properties and rules are different, and maybe that would regulate D-Serine release differently as well. It is. It is. I wish I knew. This is something that's puzzled me as well, and a few reviewers too, actually. This is not the first time that this is observed. There is a lot of evidence in the literature that adenosine as well is maintained in slices at some levels. Even acetylcholine, in fact, there's a few labs that show that if you just block cholinergic receptors in slices, you do see an effect, which means you do have endogenous acetylcholine in the slice that escaped degradation by cholinesterase, which is puzzling, but it is a fact. And so we think that this endogenous amount of acetylcholine is somehow creating a tonus of D-Serine, release pathway. So we believe that why is the D-Serine kind of always present there in slices? Might be because there is always a little bit of acetylcholine in the slice.
It kind of related to that. I was just curious, your model still has an astrocytic process near a synapse.
Mm-hmm.
It sounds like it doesn't really have to be the way that D-Serine might work. It might be not required locally, but a more global signal, which could explain why not all synapses might have an astrocytic process.
So we do believe that D-Serine release is extremely local, and I didn't have time to go into this, but that's what we showed during my PhD, that as soon as you go outside of the synapse and look at extrasynaptic NMDA receptors, they are not gated by D-Serine at all. They are gated by glycine, actually. So those receptors probably don't even see D-Serine. We believe that there is an uptake system near the synapse, either by neurons or astrocytes, that assures that D-Serine stays within the synapse and doesn't really spill over outside.
In the interest of time, Thomas, going to be here to answer a few more questions from John tomorrow. So thank you.
Thank you.